Good afternoon, everybody. Thanks for joining us for this lecture, the latest in our, I think it's our fourth season of lectures. We're in for a real treat today. We have with us Dr. Chris Morris, who's Senior Lecturer at Newcastle University, and he works in the Translational and Clinical Research Institute. Chris's two areas of focus are on neurodegenerative disorders like Parkinson's and Alzheimer's, and in neurotoxicology, in identifying how different poisons may affect the nervous system. We've supported Chris's work for, I don't know, it must be best part of 10 years now. And Chris's work is looking to identify a way to see whether somebody's been exposed to carbon monoxide by looking at the changes in white blood cells. I won't describe how he does that because I'm sure he will cover that in his presentation. And Chris has been a member of the British Toxicological Society for many years and has sat as a committee member on the UK government's DEFRA committee on pesticides. Since 2013, Chris has been external chair of Parkinson's UK and the Multiple Sclerosis Society Brain Bank's peer review panel and a member of the Parkinson UK College of Experts. So that's Chris. Is Chris in the room with me? I can't see, but I'd like to hand over to Chris now. I was going to wait for Chris to come into the room. There we go. Hi. So there we go. Thank you, Chris.
Thanks for the invitation here, Adrian, and thanks to Lauren and Kimberly at Court for looking after me so well in the run-up to this. So, as Adrian has mentioned, we've been funded by the Carbon Monoxide Research Trust for a little while now, looking at alternatives to carboxyhemoglobin as a marker for carbon monoxide exposure. And we've been doing that a long while, trying to find something that gives us a little bit of an alternative or at least an extra add-on to measuring carboxyhemoglobin in the blood. Let me just go to full screen. So, bit of a prosaic background. We've known for rather a long time now that carboxyhemoglobin, or rather carbon monoxide, poses a problem in terms of exposure and in what it can do. And I think one of the salient points from this little piece here is that often as not, carbon monoxide exposure and the dangers of it are simply put to one side. So in the case of here, this little piece from Thomas Hardy, we've got somebody at the end of the day, they come back from work, they're tired, they've got a headache, and they simply lie down. And all too often we see the consequences of that can be very, very severe for an individual. And it's still happening now, where we see perhaps 20 to 40 deaths per year just in the UK, much more widespread, the problems of carbon monoxide poisoning. Hospital exposures, we're seeing thousands, perhaps as many as 25,000 exposures per year just in the UK. And obviously, if you go to different countries who use different solid fuels, because it is typically solid fuels that are causing the issues, or at least carbon-based fuels, we see lots of these cases turning up and causing issues throughout. And I think that previous little piece that we saw there was showing us that it is difficult to diagnose carbon monoxide exposure. People don't understand, certainly, what the general issues are around it, that it's a very vague kind of experience that people have when they're exposed to carbon monoxide. The diagnosis is problematic. It's not simple. People come in with, particularly at this time of year, colds, flus, they've got headache, they're tired, etc., and they just put it down to something else, where in reality it could be carbon monoxide exposure. So what we need to do is work out ways whereby we can accurately figure out if somebody has been exposed to carbon monoxide, and then put in place preventive measures that will help people steer themselves away from the dangers of carbon monoxide, and in future, perhaps even eliminate it altogether. So the problem with carbon monoxide is that it's a very reactive molecule. It reacts with different compounds in the body, causes those irreversible changes, and leads to these non-specific diagnostic features that you can see with carbon monoxide exposure. And it's really to do with its affinity for heme groups. And we normally, when we think of heme groups, we think of the blood transport protein hemoglobin found in red cells, and that's where the bulk of heme is. Normally it's carrying oxygen, but when carbon monoxide comes along, maybe it's a faulty boiler, a blocked flue somewhere. When we're exposed to carbon monoxide, we breathe that in, it goes into the body, and it displaces essentially oxygen from different heme groups, and particularly from hemoglobin and other heme-containing compounds. And we've got this very, very high affinity for carbon monoxide for these different proteins. So it's stopping those proteins working. And although it's reacting lots with hemoglobin itself, it does react with a whole load of other compounds as well. So things like nitric oxide synthase there, NOS, COX, which is cytochrome oxidase, and other different cytochromes within the body which contain these heme groups. This is what carbon monoxide targets. And there are two essential effects that it has on the body. First thing it does, because it's getting rid of oxygen from our red blood cells, it's causing anoxia, in other words, a lack of oxygen to the different tissues that we have. And because we use that oxygen to generate energy, to drive all our cells and all our physiological processes, we're causing energy depletion as well. So we've got a two-hit problem here. And naturally, the two organs of the body that use oxygen the most are the heart and the brain. Heart pumping that blood around 24 hours a day, and the brain using up lots of oxygen, requiring lots of energy for all of our different processes throughout the body. So that's where we see the main effects of carbon monoxide. For the brain itself, and this is the bit that I'm interested in particularly, we have several problems. And this is just a picture of somebody who's been sadly exposed to very high levels of carbon monoxide and the damage that it causes in the brain. We get this thing called bilateral striatal necrosis. It sounds really, really horrible, and it is. We get a Parkinsonian, sort of get a clinical syndrome that looks very similar to Parkinson's disease caused by carbon monoxide exposure. We get problems with an area of the brain called the hippocampus, which is involved in learning and memory. And so you can get amnestic memory loss, problems with thinking as well. And we get these different things. You can get strokes with it, but you can also get damage to the back of the brain. And so you get problems with walking and fine movement, this ataxia type of problem. So for the brain itself, very marked changes can be caused by very high levels of exposure. And we don't really know itself what low levels of carbon monoxide exposure might do to the brain, chipping away slowly at different brain processes that could be occurring as well. And for the heart, again, if somebody has a predisposing heart condition, if you're stopping oxygen getting to the heart itself, reducing the energy, we can cause very severe heart problems in people who have pre-existing conditions as well. So one of the things that we've done to try and see if we can see what carbon monoxide does is we've looked at people who have been exposed and sadly died due to carbon monoxide exposure. And again, because I'm interested in the brain, we've looked at why the brain might be particularly vulnerable along with the heart. And so we've looked at one of the key enzymes involved in brain function, and this is complex 4 of the mitochondrial respiratory chain. So the mitochondria, these little batteries that are in virtually all the cells in the body that generate lots of energy. And we wanted to see, because it's got lots of iron in this cytochrome oxidase, does carbon monoxide affect it? And it turns out it doesn't really affect it here. And so probably what we're seeing for lots of the effects of carbon monoxide on the brain and probably on the heart as well is purely that you're getting rid of oxygen. You're stopping the oxygen getting to these vital organs. And that's how carbon monoxide's having its very serious effects on brain function, heart function, and other parts of the body. We have a marker for carbon monoxide exposure, and this is carboxyhemoglobin. So that's typically what people will look for to try and see if somebody has been exposed to carbon monoxide and see if somebody who has any of these particular problems, who's been within a situation where we think there might be high levels of carbon monoxide, this is the marker in the body that we're looking for. So this is where, instead of oxygen on hemoglobin, we've got carbon monoxide on hemoglobin. Now, if you look at the average person, they have around about 0.5% of carboxyhemoglobin in their bloodstream, maybe 1 or 2% at the outside. But if we look at a pregnant woman, because the pregnant woman is breaking down some of our own stores of heme supplies to provide the baby with iron while it's developing in the womb, carboxyhemoglobin can go up to about 5%. And if we think about smokers as well, so cigarette smokers in particular, because they're combusting cigarette nicotine, the tobacco, they're generating lots of carbon monoxide as part of that process. That's getting into their bloodstream through their lungs. And their carboxyhemoglobin can be 3 to even 15% of their hemoglobin in the body being carboxyhemoglobin. So sometimes you can get very high levels of carboxyhemoglobin. This poses a bit of a problem in that if somebody is looking for carbon monoxide exposure, you're going to measure carboxyhemoglobin, but instead of looking at that 1 or 2% in the normal person, the background is that perhaps you're looking for anything more than 10%, because somebody who could, the person who you're looking at could have come in and they smoke, and therefore, if they've got a high level of carboxyhemoglobin, 10%, let's say, that's perhaps average for somebody who's a smoker. And you might not therefore think that the person has been exposed to carbon monoxide. So when in a clinic carboxyhemoglobin is being measured, there's a threshold that it has to pass before a clinician, a doctor, will think, oh, this person might have been exposed to carbon monoxide. There's also another problem as well. If you remove a person from being exposed to carbon monoxide, the carbon monoxide in the blood rapidly drops, and it takes roughly 30 to 80 minutes for it to go from, let's say, 20% to 10%. So within a very short period of time, unless you think there's a problem with carbon monoxide exposure, you can get below the threshold where somebody thinks there's carbon monoxide. And remember, at the background of this, there's somebody who's poorly, they've got headache, they've got fatigue, they're not breathing particularly well. Is that because it's carbon monoxide exposure, or is it because there's something else going on? And so often as not, carboxyhemoglobin isn't even measured. So we've got a situation here whereby we've got exposure to carbon monoxide, but what we typically use to measure carbon monoxide exposure, carboxyhemoglobin, has to be very high, and we have to get to it relatively quickly. And this is illustrated by this thing here. So imagine somebody here is exposed in their home to high levels of carbon monoxide. And here they've got 25% carbon monoxide in their blood when they're poorly. The paramedics arrive, they take them into the ambulance, and within perhaps half an hour, it's dropped down to 13%, which is very close to what our smokers would have. If you're putting an oxygen mask on because the person's not breathing, they're not really very well, that will drop even quicker. And so by the time they've moved themselves on, got to accident and emergency, we might be down to 10%. That's certainly near our threshold. If we get any further, 90 minutes, not unusual, sadly, we're down to 5% carboxyhemoglobin, and nobody might think, even if they were measuring it, that that is a problem. And back at the home, there is a problem with carbon monoxide exposure. So we have a marker here, although it's good, it disappears quickly, and we might not think about measuring it. So what we might need is another way of detecting carbon monoxide exposure. There is a problem. Although we have carboxyhemoglobin in the blood, I've probably got it, that baseline level comes from within us. So constantly throughout life, all of our hemes that we produce for making red blood cells are being broken down. And they're being broken down by this enzyme heme oxygenase, which is producing, in an average adult, about 14 milligrams of carbon monoxide every day. So we have this background level of it. It's not necessarily bad. That amount of carbon monoxide that's produced within a cell, within a body, in my body, in everybody's body, is doing lots of different things that are of benefit for different cells. So we know that carbon monoxide, if it's produced in those small normal amounts, it will help prevent damage to people's DNA. So it helps with cancer prevention. It helps to divide our energy-producing mitochondria. It prevents some inflammatory responses that give us things like rheumatoid arthritis and so on. And it stops cells dying. So it has its good benefits. So if we think about that, can we measure those good benefits and some of the ways in which it protects cells? And we've done that. We've looked at the brains of those people who sadly have died from carbon monoxide exposure, and we do see changes to some of the proteins that get involved in the normal physiological role of carbon monoxide. And that's this SGCB1, easy for me to say, P38 MAP kinase. We see changes in the levels of those, which tend to tell us that if somebody's exposed to carbon monoxide or is producing carbon monoxide, then you can spot these physiological responses. So that's given us the idea that although carboxyhemoglobin has its uses, and you can detect high levels of exposure of carbon monoxide, that there could be other ways of measuring carbon monoxide exposure. And so we've tried to set out to look at that. We've given ourselves a bit of a challenge in that we need a carbon monoxide biomarker. A biomarker is just a posh word for something that tells you a particular process is going on. In this case, we need to know about, is somebody being exposed to carbon monoxide? We need to have a long half-life. In other words, that marker needs to hang around long enough that even if the carbon monoxide is gone, if we remember back to our home, to ambulance, to hospital scene, it's disappeared very quickly from the blood, carboxyhemoglobin. We need to be better than that. So we need to have something that's relatively stable. Even if somebody's been exposed a little while ago, it needs to hang around so we can measure it. We need to figure out whether the carbon monoxide response that we're going to measure gives us a measure of how much exposure to carbon monoxide has occurred. So it has to be dose dependent. We have to measure those. We also have to make sure it's very sensitive and specific. So we can't have other compounds that enter the body or are produced, or different conditions, different disorders that somebody might have. Perhaps they've got diabetes and some markers change. We can't have that interfering with the process of measuring something that's specific for carbon monoxide exposure. So we have to have the sensitivity and the specificity of measuring. It has to just measure carbon monoxide exposure. It's got to be quick and easy. Carboxyhemoglobin's quick and easy. We also have little finger monitors that will do that very easily. But again, that depends on how much you've got in the blood. If we can make it as non-invasive as possible, then it's very, very simple to use. But it has to be specific for carbon monoxide. So we've set about doing that. We've set about trying to give ourselves this challenge of finding something that measures carbon monoxide exposure. And it's that physiological change to carbon monoxide that we're starting to investigate. Can we use that normal system that the body produces carbon monoxide, then the body responds to carbon monoxide, to identify a marker that will tell us somebody has been exposed to high levels of carbon monoxide? So we've done that in 2 stages, and I'll talk about that now. The first thing we've done is we've taken cells and we've exposed them to carbon monoxide to see what happens. Simply, what changes occur within a cell when you expose it to relatively high levels of carbon monoxide. So we've done that by taking volunteers. We've asked them for a blood sample, white blood cells, find them circulating in their bloodstream all the time. We've taken those peripheral blood mononuclear cells, the PBMCs. We've isolated them. We've then exposed them to a relatively high level of carbon monoxide in air. And it's very similar to perhaps the levels that you would achieve if the flue was blocked on a gas boiler or something was wrong in a chimney and it occurred. And we're kind of thinking here, well, it's a wintry day, a little bit like we have now. From 5 o'clock in the afternoon, the boiler comes on, the carbon monoxide starts to build, and 11 o'clock at night, when everybody's off to bed, it stops. So the exposure is about 6 hours. So we've taken our cells, we've exposed them to this 200 part per million in air, so they're still getting oxygen, they've still got everything they need to survive, and we've taken them there. Then what we've done is we've taken those cells at the end of that exposure and we've measured all the different changes in expression of all the different genes that occur within those blood cells. And we've tried to look at what changes occur there. So this is our very high-tech exposure chambers. Some of you might see these are these clippy boxes that you keep your sandwiches in when you come to lunch, but these are very large ones. Very good because they have a very nice gas-tight seal in them. We have these inlet and outlet ports. We have a fan on the top that moves the air or the carbon monoxide filled air around. We have these, which are party balloons. Yes, you can see red and gold party balloons on here. They allow the gas to expand because when we push it in, it's slightly cooler. As it warms up, it expands when we close the box. We put our cells in there, we expose them for the 6 hours, and then we look at the gene expression changes. So that's our high-tech kit. We see lots of changes occurring. So we know, we think we know, these are changes which carbon monoxide seems to cause to these white blood cells. And we're using the white blood cells simply because they're easily accessible. And if you took a blood sample from somebody who you think might be exposed to carbon monoxide, maybe those blood changes that we're seeing in the blood would reflect what we see in our cells exposed here under our ideal laboratory conditions. So we see changes in mitochondrial genes, and remember that our mitochondria are targets of carbon monoxide. We see changes in the energy producing final part. We see changes in COX, so this is one of the known targets of it. We see it in complex 2, which is a little bit unusual in that it's not an iron-containing one. So we think we've got something going on here. We see a signaling pathway as well. And one of the important things about this signaling pathway is at the end of it is nitric oxide. So again, this is another target of carbon monoxide, one of the well-known targets of carbon monoxide. So we know now that we're getting these positive responses by these little cells in these little incubation chambers to the carbon monoxide, and they're changing their expression of different things. And the other thing that we get as well is this signaling pathway. We're working on white blood cells here. They're the ones we're going to see if we're in circulation. And we see particularly targeting this area here. We know that these enzymes, NOx and COHb, they have, again, heme groups that would typically be targeted by carbon monoxide. And perhaps this change here that we're seeing is one of the reasons, particularly in smokers, why they often get changes to their blood vessels. So this is a blood vessel here. This is the actual white blood cell. And so we're seeing changes happening there, which again might relate to carbon monoxide exposure. So from all of this, we're seeing changes which are probably physiological, but now they've turned into pathological changes that occur when we've got high levels of exposure to carbon monoxide. Question is, can we take those changes in expression of these different genes in our white blood cells and turn those into a test? So we're seeing these cellular changes in physiology. We think these are the pathological versions of it. So they're affecting systems that would normally perhaps be affected by endogenous carbon monoxide, but they've now gone supernormal. We now need to ask the question, though, is how long do these changes last? Because they could only be there for a very brief moment and then disappear. Question is as well, are these meaningful? Do they tell us something about how carbon monoxide has its effects? And does it mean something in terms of what we know about carbon monoxide as well? Can we turn them into a test? And one of the problems that we have here is our smokers. Remember that they've got very high levels of carboxyhemoglobin normally. So we now need to try and test these different changes in smokers. So what we've done now is we've limited ourselves to just a few of these changes, perhaps the important ones, the ones that are meaningful in terms of physiology, meaningful in terms of what attacks, what carbon monoxide attacks within the body. And we're going to now look at these in smokers as well as non-smokers. Because if we can see the changes in smokers, then we've got an idea that this will work no matter what. It'll work in people who have been smoking, where we know carboxyhemoglobin's not that effective, but certainly below a certain level. If we can get that change in the smokers as well as non-smokers, then we potentially have a new test. So we've essentially done it all again. We've taken our white blood cells, our PBMCs, we've used our 200 parts per million. We've also used 50 parts per million as well. We've exposed them and we've said, can we repeat everything? Because that's one thing we need to know is all of this repeatable? We've then let the cells recover. So we've put them back into normal air, normal oxygen. And after 1 hour, 4 hours, 8 hours, and essentially overnight, we've gone back and said, are those changes in those white blood cells still there? Smokers and non-smokers, and we've used this method, real-time PCR, to actually detect what these different changes are. Now, a brief pause for statistics. Some people might remember the film 2001: Space Odyssey. This is a bit like me going to the statistician. And in 2001: Space Odyssey, there's this big, super intelligent obelisk, and then there's the ape creatures. And I'll give you no guess as to which one I am when I go see the statistician. He's the obelisk. So I go to Steve. Steve weaves his magic and tells me something's going on if I take this particular gene set. And so if you look at these different gene expression changes, they start to cluster into little areas, which tell us that certain processes are being affected in the white blood cells by the carbon monoxide exposure. So we've got this big area here, smaller one out here, maybe a one in the middle here, and something out at the top. So again, using all of these different approaches, these statistical methods, which are well beyond me, we can find certain genes that seem to change in response in these white blood cells. And some of them seem to be independent of whether the person smoked or not, so that they would cover everything and anybody, perhaps, who's been exposed to carbon monoxide. And there are a set there, and I've kind of broken them down into 3 different areas. This one up here, the green one in the middle, which is perhaps the most important one, and then this red one, where we can see changes that are almost specific to smokers. So this is the first set. Wonderful name, hypoxia-inducible domain family member 1A. It's really involved in the response to oxygen and energy levels. So if you imagine a cell, it needs to get oxygen in, it needs that oxygen to help in production of energy, so it'll drive all of those cellular responses. And what it does, this particular protein, HIGD1A, is it helps to produce our mitochondrial respiratory chain. So all of the little enzymes within these batteries of the cell that generate all the energy. So again, we know that part of carbon monoxide's dangers is that it reduces the oxygen level. It pushes oxygen out of the way. So we think what's happening here is we're detecting this particular gene changing in response to carbon monoxide pushing oxygen out of the way. And we're seeing a similar thing here. This again, wonderful long name, PRKAG1, used to be called AMPK gamma. Again, this protein itself regulates lots and lots of different cell processes. It tells cells what to do in response to certain things. But again, we think here there's a cell stress. Essentially, that carbon monoxide is causing stress upon the cell. They're not getting enough oxygen usage, they're not producing enough energy, and so things are changing. But one of the key things that happens here is that this AMPK gamma changes the expression of nitric oxide synthase. So this is a key enzyme that we know gets affected by carbon monoxide. So we think again, this might be changing in response to our carbon monoxide, and it has perhaps a physiological relevance and a pathological relevance as well. And then we come to that big group in the middle. And the first thing we see changing is nitric oxide synthase. We see these particular changes. We know that nitric oxide itself is an enzyme that's important in carbon monoxide, probably because some of the early effects of carbon monoxide exposure, the headache that accompanies carbon monoxide poisoning, is probably caused by nitric oxide signaling. So this is a vasorelaxant. If you don't have enough of it, you get vasoconstriction and the headache. But what we're seeing here, which is quite important, is that after the first exposure that the levels drop, but after an hour and even after 18 hours of exposure, the levels seem to have dropped and stayed low. So even when the carbon monoxide's gone away, the cell still thinks there's a problem. And so this might be a useful marker that tells you, even if a little while ago, somebody was exposed to carbon monoxide. And we know it's a target of carbon monoxide exposure as well. So that's important in giving us relevance to what we're finding here. And this is another one. This is guanylate cyclase. So this is another well-known target of carbon monoxide. We don't really see a change, or at least not a superbly significant change at 6 hours when we've got maximum levels of exposure. But certainly at 1 hour, we see drops in the level of this enzyme, which kind of parallel what happens with nitric oxide. And we do see similar changes at 18 hours, although it doesn't seem significant in the small group of individuals that we've looked at. And just thinking back to those individuals who sadly died from carbon monoxide exposure, we see similar effects in them, in that their levels of the protein itself dropped in the brain. So we think this is a pathological response. We've got some backup for this changing in the brains of people who've been exposed. And again, this is part of the pathway that our previous enzyme PRKG was involved with. So it's all part of this response system to carbon monoxide. Then we have this other one, F3. F3 in itself doesn't seem a very good name. It's a very short name, but it used to be called coagulation factor 3 or tissue factor. So it's very important in blood coagulation cascade. What it does seem to fit in with is this protein here, VEGF, vascular endothelial growth factor. It's very important in modeling and making and maintaining blood vessels, and particularly blood vessels in the brain. It is also found on the surface of white blood cells, so we know it's in our little white blood cells that we've been exposing. But this protein seems to be activated by VEGF, and potentially it responds through a different pathway, particularly this ACT pathway, to produce VEGF within the cells, and this causes some kind of a feedback loop, possibly from carbon monoxide. We need to look into that a little bit further though. But certainly there are changes here that suggest it doesn't matter whether you're a smoker or a non-smoker, that these changes are occurring. And again, this one here, this seems to be quite a robust change. Again, we know that this receptor type 2 responds in some ways to nitric oxide, so it's all coming back to that, coming back to the guanylate cyclase, that these changes are taking place, maybe altering the way the white blood cells are shaped and how they interact with blood vessels, because we know that in the smokers, that if you get these changes in white blood cells, they tend to be much more sticky towards blood vessels, which is why you get the vascular problems in smokers. This is peculiar in that it's mostly smoker related. So this change in the vascular endothelial growth factor seems to take place, seems to be altered. We know what this does, but again, we're probably seeing a supernormal, a pathological response to carbon monoxide that we'd see, again, which eventually leads to nitric oxide production. So again, it's this kind of loop system that we're seeing, carbon monoxide exposure affecting nitric oxide and then looping back and around. So this seems to be something there. We're not sure here whether the smokers are primed to respond to carbon monoxide in an abnormal way, maybe because they've got changes in their white blood cells, because they've been exposed to carbon monoxide a very long time, that when they get a zip of it, things start to alter very markedly. But it seems to be pushed towards the smokers as well. So we've got a series of markers here that we've identified in a first round. It seems that we can see changes again when we've repeated the experiments, we've extended the experiments in different people. We've used smokers as well to make sure that we can cover as broad a population as possible. And we think those changes seem to relate to the normal physiological production of carbon monoxide within the body, but because we're exposing to high levels, we're getting a pathological response here, some of which, particularly in the case of NOS3, it seems to last for a very long time, and maybe in the guanylate cyclase as well. We've got cell pathways that seem to be indirect, and maybe they're due to the energy depletion that's being caused. And we might get some of these changes that are specific to smokers as well. The next steps. What do we need to do next? It's great because we have the Carbon Monoxide Research Trust, and it's always about joined up thinking. It's about how do you take something that's in the lab, which is slightly unusual, and can we use that to get out to people who are exposed to carbon monoxide to say there is a problem. And I like the way that COHb thinks here. So I think our next steps with what our study is telling us is we need clinical samples. And just to give it a nice little pun, those clinical samples are as rare as your average hen's teeth. So I think we need now to go from lab to clinical samples. Fortunately, because of joined up thinking, we're working with Sheffield Hallam University with Marie Herigstad and her group there. We'll be able to obtain clinical samples from people who have been under very controlled conditions exposed to small, low levels of carbon monoxide. And so the answer here would be, can we see those changes that we see in the lab occurring in those exposed individuals under very controlled circumstances? Be great if we could. We also need to take it out even further. Can we do a more, let's call it community-based study? Can we obtain samples from people who might have been exposed to carbon monoxide, whether we've got a measure of carboxyhemoglobin, either at a baseline level or further down the line? And can we take those samples? Can we relate any changes that we might see in our measures? And can we relate those to carboxyhemoglobin? Do they also last as well? We've seen with nitric oxide that the gene expression changes lasts for certainly overnight. Can we see the changes even if they've been removed from an exposure to carbon monoxide? And do they work with high levels of exposure? Do they work with very low levels of exposure? So we can figure out what even low levels of exposure to carbon monoxide might be doing. So that's our next steps. We've got to take our findings here of all these different gene expression changes that seem to make sense in terms of carbon monoxide exposure. We've got to get those clinical samples through our research network that CORT has set up. Can we use those? Can we take this little set of gene expression changes and can we turn them into an alternative biomarker for carbon monoxide exposure? So I'll stop there and say thanks to the people who actually do all of the work. My team here in Newcastle, Medea, Leona, John, Carmen, Peter, Lena, and Steve, the Obelisk 2001 statistician who does all of the magic and turns it. And always to Isabella, Izzy Myers, who gives us loads and loads of encouragement and sends us in the right direction as well. So I'll stop there and say thank you for listening. So handing back to Adrian.
Thanks, Chris. That was a really, really great presentation. And I particularly like the Thomas Hardy reference and the 2001 Space Oddity reference. So we've had quite a few questions in the chat. And we've had a question from Dr. Mari Herigstad, who leads the Sheffield Hallam CO research group up there in Sheffield. And she asks, in the event of us finding a new biomarker and validating it, what does Chris think are the obstacles for it being adopted in, say, the NHS?
Ooh, obstacles for taking it any further forward. Um, it's got to be a lot better than carboxyhemoglobin measurement. We know carboxyhemoglobin measurement is good if you've got high levels of exposure. We've got to make sure that it gives us an adequate reflection of exposure levels. So if we see a very large gene expression change in some of these different genes and we put them all together, is it going to tell us about the levels of exposure? So I think it's got to pass muster in that way. I think once it's done that, big enough samples, big enough patient population to convince the NHS to say this is worthy to use a bit further, and then taking that set of markers and putting it into a small scale study that says if we have somebody coming in with these vague symptoms, and that's one of the problems about carbon monoxide exposures, these vague symptoms that people come in with, still very debilitating. Can we identify those individuals who have particular exposures? Is there a subset of those individuals that we can spot? And that's how we take it forward, by doing good, well-powered scientific studies that we can then take to the NHS and say, this is what you need to have in maybe an accident and emergency clinic. So you can use this as a test for exposures.
Great. Well, thank you. We've got two questions from David Wood, who is at St Thomas's. The first question is, what is the response change in white blood cell markers in the setting of mixed exposures? For example, house fires, where there may be things other than carbon monoxide.
We haven't done that ourselves, but I would be thinking that similar things could occur, certainly down the kind of lack of oxygen route. One of the things that you're going to get produced there is going to be carbon monoxide, but you're also going to get things like cyanides produced, which have a similar effect, but they wouldn't necessarily affect things like nitric oxide. So that is potentially one extension of the experimental system that we do now, in that we could put small levels of that in and just see if that causes any problems and just tells us, well, we can't take it any further. But I don't think that the nitric oxide system gets affected by cyanide directly. So this is certainly a very good question, certainly something we would need to test on the route to producing a good biomarker.
Great, thank you. And David's other question is amlodipine activates endothelial nitric oxide synthase 3. Does amlodipine have any other actions on the white blood cell, NOS3, that would impact on the use of this biomarker in clinical practice, given how widely amlodipine is used in the management of blood pressure?
Yeah. And again, it comes back to some of the older patients who might have kind of pre-existing heart disease and how that affects them. The answer is, straight off the top of my head, I don't know what amlodipine might do to these things, but certainly we'd have to test a lot more conditions to see how robust these markers are under different circumstances. Again, as we see with the smokers, that there's something in the background that might alter their VEGF system. But certainly the other markers that we see seem to be independent of those. So yeah, certainly some things that we can put into the mix and test to see what might cause us any problems in the biomarker system.
Great, thank you. Another question here from Leo Otterbein. He says, great talk. Do you know if the gene expression changes correlate with protein expression and activity of those genes? Have you correlated your data sets with the RNA sequence studies that have been reported in humans exposed to pure CO, i.e. remove the complexity of smoking, which contains many carcinogens? Sorry.
Yeah, I mean, I guess we've got no carcinogens, we hope, in the system that we're looking at in terms of the smokers. We haven't correlated protein changes yet. I mean, the nearest one we got, which I mentioned there, was the guanylate cyclase, in that we see drops in the gene expression for guanylate cyclase. We see that in the brain of people exposed to carbon monoxide. That would be another step in the process. Obviously, gene expression is before protein changes. Sometimes these protein changes don't translate to a gene expression change. We've done that a lot in the past and seen these things. Again, it's something to bear in mind. Do you go after the gene expression change, which we can see quite happily there, or do you go for the protein, which may take a longer time to start to change? It's one of those things you have to add to the mix to see if you can get the most robust answer, but certainly something we need to look at.
Great, thank you. Got a couple of questions from Albert Donne, who is a consultant toxicologist based in the US. I'm not sure, like, I'll ask the questions, but I'm not sure how specifically to this particular project. Did Dr. Morris consider exhaled CO testing, which is of course 100% sensitive and specific, as well as non-invasive, reproducible, and correlated with severity, etc.? And unlike testing arterial or venous blood, breath CO tests can distinguish CO levels diffusing from lungs and the average of all tissues. I mean,
I think that's a very good question. I think exhaled CO is a very good measure. It's very like carboxyhemoglobin. I think the validity of that is, again, you need to get in there quite early when you've got a relatively high level exposure, because the more you exhale, the less that breath is going to have in it to measure. Yeah. So you will get below measurable levels relatively quickly. So I think, though, that's a good adjunct, or maybe supersedes even carboxyhemoglobin measurement. But it's thinking about when you've been 4 or 5 hours down the line and that exhaled CO has gone completely, your carboxyhemoglobin is back to normal levels, your exhaled CO is back to normal levels. That's the kind of area that we need, where we can look in an accident and emergency department, which is very busy. Can we get those individuals and say, yeah, 6 hours ago, you were exposed to too high levels of carbon monoxide, and that's why you've ended up here.
Thank you. So I'm just looking at the other comments. Albert's shared the article that Professor Rose, I think he's based in Pittsburgh, put out in 2019 in Critical Care magazine. I think we can share that link when we circulate the link to the presentation afterwards. That was quite an interesting article that came out, was it now 6 or 7 years ago? I'm pretty sure that you'll have seen that, Chris. I've brought Izzy into the room here because Izzy's asked a couple of questions, and of course she's been involved in this work and a number of other projects that we've been involved in. So rather than me read out her questions, I thought I'd put her on the spot and bring her in the room and let her ask the questions herself. I hope that's okay, Izzy.
Yes, that's fine. I mean, it might just show me up to say that I don't know what's going on.
No. Say it.
So.
Not at all. No. I'm just gonna really say that we're doing some work with medical examiners and e-learning, and with that in mind, you were mentioning some of the results that could be quite useful for pathologists, perhaps, rather than using COHb as a marker. But I don't know whether the method of extracting what you found is harder than measuring the COHb in the deceased rather than those methods that you utilized?
Yeah, I guess there's two approaches. If you've got somebody who has sadly died while still exposed to carbon monoxide, then COHb is typically always high. It's relatively straightforward to measure.
Stable, yeah.
You could have that as a, well, we know that's high, and we've now checked this, it's still high. So it would be something parallel that would kind of add additional weight, I guess.
Yeah.
So
But
yeah,
not necessarily to tell us any more. It's still that yes, no
yeah,
sort of answer, isn't it?
Yes.
Yeah. And my other question was just regarding the work at with Sheffield Hallam, and it's because I can't quite remember, so apologies. Was whilst having those exposed to controlled amounts of CO, are we also looking at those who are also smokers as well? I mean, it's quite it's such an odd area, because usually that idea of not engaging with those areas that could cause confounding, but obviously smokers are exposed to environmental CO as well, and they need to be taken into account. And you found some interesting stuff with the smokers, so I was wondering how we're going to what's going on there.
Well, I think in that instance of looking at the smokers, it was to make sure that we had a marker or a set of markers that it wouldn't matter what the person had been exposed to constantly, we'd still be able to see some changes that are caused by acute exposures to high levels of carbon monoxide. And that's the problem with, I guess, carboxyhemoglobin. You've obviously got this very high level of carboxyhemoglobin in the smokers, which confounds things. But let's take all of that on board and everything we know about smoking and say, right, well, we're still going to have to include those people. They still might be exposed to acutely high levels of CO. So bring them in. Let's see if that causes any problems. And if we can get around that, and it looks as though things are going around that, the treatment effects are there, then we can take that biomarker set a little bit further.
Yeah, that's great. Thank you very much.
Thanks, Izzy, and thanks, Chris.
Lovely to see you as well.
Yes, likewise.
Brilliant. Well, thank you. We're now coming up to 3 o'clock UK time, so we're about to draw this to a close. Thanks to Albert for his comments and for sharing that link. He just wanted to point out that the work that the person who undertook the study he mentioned was undertaken by Dr. Jason Rose, who apparently now is at the University of Maryland. And finally, we had a comment from Jordi de Sola, who is in Barcelona. He concurs with the high value of CO in expired air as the most sensitive marker for CO poisoning, and he's undertaken a critical analysis of more than 4,000 cases. That's prospectively analysed. Jordi has been a participant in our previous incarnation, the Gas Safety Trust. He participated in an event at the Royal Society of Medicine some years ago now. And thanks for that comment, Jordi, and I hope you'll continue to stay involved with us. It'd be good to talk to you about the work that you've been doing there in Spain. So I think do you have any final comments, Chris, before I thank everybody and let them get on with their day?
No, just once again, thank you, Adrian, for organising this. Thanks to Kimberly and Lauren as well. And good luck.
Thank you. I can't really claim much credit for organising this. The hard work has been done by Kimberly and by Lauren. And so thanks to you, Chris, for presenting today. It's really fascinating, and I think there are quite a few things I'd like to pick up on with you and look at how we move these things forward and you're aware of the other work that we're supporting in other parts of the country. And perhaps we can try and join the dots a little bit. I was about to get out. I almost got out, but Izzy's
I
put
know.
her hand.
I just wanted to say, and I'm sorry for butting in, but just to acknowledge that Chris Morris's work was probably the first piece of truly toxicological work that the CO Research Trust, then Gas Safety Trust, ever actually funded, and but probably back in about 2015. So
Yeah.
I just really wanted to say, from my perspective, how wonderful it is to hear how far the CO Research Trust has come in its endeavours to support really great basic scientific research. And I think what Chris has shown us today has really been the start of something that was some time ago, but really was a fundamental change in the way that the CO Research Trust looked at funding research. So congratulations to Chris in instigating this change and for this great lecture today.
Thank you.
Thank you.
Thanks, Izzy. That's nice to hear. And I think that it's good for the board members, our trustees who are on the call, to hear that too, because it's their enthusiasm and I guess bravery in changing the direction of the sort of research that we funded that have led us to this point. And of course, we can't do it without all the smart guys in the room doing the hard work. Anyway, it's 3 minutes past 3 now. So I'd like to thank Chris for his lecture today. Thank everybody who's asked the questions and the comments, and thank all of you who have attended. We will send a follow-up email after this to everybody who signed up, so you'll get a copy of the presentation, so you can share that far and wide if you wish. Next month. Well, no, it's not next month, is it? I think it's May, early May, because of Easter. We're not doing something in April. We've got the Sheffield Hallam CO Research Group coming to talk about the work that's taking place there up in Sheffield. There's a number of researchers working on a number of topics there, so it'll be a really interesting project. It's the 2nd of April. Sorry, thank you, Lauren. I am on top of these things, but time does fly. So thanks, everybody. Have a great day. And thanks to all of you who joined here in the UK and overseas. Cheers. Thank you. Bye-bye.
Thanks, everybody.